Good afternoon to my channel, Science and Vighyaan. Today, I'll discuss the important web server that is free of cost. You can use it for the ligand-based study and the structure-based study. You can see I have already opened the Google. Fine. Generally, we waste our time by browsing the Google uselessly. But today, I'll tell you the important feature of some web server or the website that are very helpful for the doing drug designing. Fine. Okay. In the Google, you have to just type down when you have a free time and there is nothing to do. What you can do, you can just open the Google browser and type here PDBSUM. Fine. You can see there are the different uh, searches found. Fine. We have to open. You can see on the right side. On the right side, there is a PDBSUM. Before going to the website, you should know what do you mean by the PDBSUM? PDBSUM, it is a kind of database. It is a kind of database that is useful for studying the macromolecule. Macromolecule is, in short, that is known as a protein. We know what are the protein. Protein are the macromolecule, and the PDBSUM website that is developed by the. You can see here it is mentioned Roman Laskowski and collaborator and the University College London. If you want to prepare any kind of competitive exam, these type of question can come. Who develop? This is especially for the bioinformatic people as well as the medicinal chemistry people, because in the medicinal chemistry, what we do, we synthesize a new compound. If we have some knowledge about the bioinformatic, we can easily design the target approach drugs or the drug that are supposed to be hit at the target. Right? Now you can see there are the different searches come. We have to open this one, European Bioinformatic Institute. Fine. That is a standalone version PDBSUM generated. Fine. Just open it, or now in the meantime, I can tell you the uses of the PDBSUM. In the previous videos, I have mentioned we have to prepare the protein. If you still have any doubt, please open my channel, and there is one playlist called computer ready drug design. You can open it, browse it, and you can find important and the very useful videos there. Fine. Okay, now we proceed to the PDBSUM. Yeah. What is the significance or the objective of the PDBSUM? Why we use the PDBSUM? PDBSUM, they will give you the idea of the protein which we obtain from the protein data bank. Fine. PDB is the protein data bank. There is one website. If you still have any kind of doubt about the PDB protein data bank and how we have to download the protein, you can mention in the comment box. I would happy to tell you about how you can download it, how you can use it for your ligand based or the structure based studies. For that, you have to now you can see PDBSUM is shown in the browser. Fine. What here we need to do? Here we do generate. There are the two versions. One is the standalone PDBSUM analysis. Either we can do, we can download, we can install it also, download also. In the next video, I'm very happy to tell you about how you can download it. Today, we will use the web server only. Today, I am not downloading anything. I just generate the PDBSUM and just tell you how you can obtain the result. Yeah. Uh, just I have clicked this one, PDBSUM generate. I just clicked it. Now it look like this. Fine. There are the two options. Uh, Either I can mention here email ID, or I can open the detailed version of the information about that PDB here. Fine. Okay. Firstly, I need to upload the PDB format file here. Fine. Or I can open this one. So what I need to do for that, I just mentioned here, I would like to install one acetylcholinesterase inhibitor, their PDB side 1E1 PDB. I just download it. Yeah. This site I need to open rcsb.org. Right. Now you can see there are the different versions. Yeah. This is a PDB website, how it look like. You can see there are the two options on to right side. Otherwise, in detail information about this PDB data bank, RCSB, I would like to uh, tell you about if you want to know about. Otherwise, this information is enough for you. What you need to do on to right side, you have to click this option. Fine. There are the different options. We need to install this PDB format option. Okay, this is the downloaded one. Now the PDB file is downloaded in my downloads. Yeah, it's look like this. And open it. You can see here I need to choose the PDB format. Yeah, sorry. PDB format. Here I need to upload the PDB format. This is the downloaded one. I just choose it. Here, if you want to obtain the result into your email ID, just mention here email ID. Fine. Okay, we'll go back. Here I put my email ID, upload. It is in the process. Yeah, it will take a time. In the meantime, I'll tell you about the PDB code. There are the different code. You can see I have mentioned here one EV. This is the protein data bank, different codes. For example, how we can assign the code. There are the there are the different classification method, trivial method, enzyme classification. With the help of enzyme classification, we have to give the name of each protein as the enzyme name. As we know, what are the enzymes? Enzymes are the itself, it is a protein, and there are families of the enzyme. Uh, in detail, there are 6 families of the enzyme. That is the reason you can see all the protein. Their first, like a first letter that is in the number only 1, 2, 3, 4, 5, 6. Fine. If you do not have idea about the enzyme, please to mention in the comment box. I would like to tell you about all the 6 enzymes that are used or that are present till now. Fine. Um, and this result will take about hours. Either if you want, I can discuss protein data bank. Okay, I can discuss about the Cocrystal and the cofactors. If in the meantime I have to obtain the result, I'll tell you. Otherwise, stay tuned to my channel. In the next video, I am very happy to show the result, how it look like, how you can retrieve the information for your papers. Like first, our first aim, why we need to do this PDB generation, why we need to study this web server. This is very important for your publication also. This is very helpful. It look like very like attractive. And when you upload any kind of paper, you can click, you can take the take the picture from this PDB some website, and you can upload the same in your paper, and you just attach it because they're very attractive, and generally editor like it. And hopefully your paper will be published very soon. Right? Okay. In the meantime, I'll tell you about what is a co-crystal and what is a cofactor and what is the difference between both. You can see this is a protein. Yeah, this one is protein. You can see, and these are the different ligands. These are either they are the co-crystal or they are cofactor. Okay, what is the co-crystal and what is the cofactor? Co-crystal, you know, co-crystal mean co-precipitation. Co mean उसके साथ आ गया कोई चीज, जैसे हम बोलते हैं को प्रेसिपिटेट, वी फॉर्म द प्रेसिपिटेट बट सम को प्रेसिपिटेट दे आर फॉर्म अलोंग विद द प्रेसिपिटेट. The same way we have to obtain the crystals, but along with the crystal, co-crystals are formed. That is the co-crystal. And cofactor, co-crystal we need it. For example, I need to work on COX inhibitor, and our enzyme is the COX, and their co-crystal is indomethacin. But along with indomethacin, some amino acid, they are still present in the protein that are not desired. Fine, that are the cofactor. I repeat, we know what is the protein. Protein is a combination of the amino acid. We know there are the two functional group, basic functional group. One is the carboxylic, other is the amino. They join together with the help of peptide bond. When we prepare any protein, sometimes protein they remain inside, unwanted amino acid they will remain inside the protein. That amino acid they are not termed as co-crystal, they are termed as cofactor, but because they are not desired one of the co-crystal. Because for example, I am making anti-inflammatory drugs, fine, but in the enzyme cofactor are present at the binding site. Now you can tell me, how could we study the uh, binding of enzyme and the ligand interaction? Because already cofactor they have taken the place of the enzyme where taken place the ligand where ligand is supposed to bind here. That is the reason when we prepare any kind of protein, we need to remove the co-crystal and we need to compare the interaction of the ligand along with the co-crystal, because co-crystal they bind to the specified particular binding site. Fine, this is the co-crystal. This one co-crystal. I'll show how you can obtain these results also. This is very easy one. You have to click here, this ball and stick. Yeah, you can see there are the different information, structure summary, then annotations, then experiment, then sequence, then genome, ligand, version. Fine, we have to press this ligand. Now, you can see this is the first cofactor. This is a cofactor. NAG to acetamido 2 dioxy beta D glucopyranose. This is a co-crystal, and these are the alignment of this co-crystal. This is the alignment of this co-crystal. Fine. Now, you can see here interaction are not shown, but we need to see the interaction. Co-crystal is binding to what site? This information we can obtain from the PDBSUM. This is the co-crystal E2020. I'll show this E2020 also to you, so that you can better idea about it. This is the NAG that I have already tell you. This is a co-crystal, fine, and this is a no, NAG is a cofactor. This is a co-crystal. Now you can see the name is 1-benzyl 4,5,4 bracket start 5,6 dimethoxy 1 indenone 2-yl methyl piperidine. Fine. Now, for example, if you are in beginning stage of your research, what you can do? You can make piperidine analogs, because you can see this co-crystal already is present in the binding site of enzyme, acetylcholinesterase enzyme. what we can do, we can start with the piperidine analogs. If you have any kind of doubt about this heterocyclic compound, like which one is the piperidine, which is the pyrazine, pyrrole, primidine, morpholine, there are the number of heterocyclic compound. If you have any doubt, do mention in the comment box. I am very happy to tell you about all the heterocyclic compound. Fine. Basically, all the drugs they are heterocyclic in nature. So see, this is this co-crystals, and they show their model complete this also model completeness also, their outlier also, everything they mention here. Now I'll show you the summary. Yeah, this is the, you can see this is the co-crystal. We can, for example, you have to work on development of acetylcholinesterase inhibitor. You can take its lead. Fine. You can make a same compound. Fine. This is the lead here. These are the hydrogens. Fine. We can see their 3D structure also. We can remove hydrogen also. Fine. Okay. In the next video, I would like to analyze the result because I think still it take times. In the meantime, stay tuned to my channel and I'll upload this video as a part 1. So please wait for the part 2 for the analysis of the result. Thank you so much. Stay tuned to my channel. Happy learning.